We really don't need that room anymore, it seems. Should we move? Anybody? So the alternative would be let me figure out whether there's an exercise. I'll check it. If we had an alternative room in the informatics building, is that what people would prefer? 1, 2. Can somebody raise the hand? You want to say something or you want to say yes? Yeah. Somebody who wants to say no. Somebody prefers this room. So it's clear. I'm going to tell you on Thursday whether we will have that room, but we will go back here on Thursday. Just because I don't know at the moment whether the room will be free on Tuesday and Thursday. All right. So today we will talk about a few, at least, words about how a structure is determined and how we can assign secondary structure from 3D structure. Just to point out, there is no lecture on Whitsun holiday, so the Whitsun holiday is Tuesday the 26th, the day after. It's not a public holiday, but it's a university holiday. Last lecture, July 7th. Oh, this is not. I have to adjust. Yes, no, this is right, July 9th. Okay, let's have the recap. What we can do in terms of predicting 3D structure through comparative modeling. So I said that if we find a sequence similarity to a protein of similar structure, and that finding is very easy here when two proteins are very similar in terms of sequence. Again, you only take their sequences, only their amino acids, you compare them using the alignment methods, dynamic programming, BLAST that I talked about. There's a realm in which this is easy, there's a realm in which this gets more complicated, twilight zone, and then it gets very complicated. And I also told you why this is so complicated, because most proteins of similar structure actually have very, very little sequence identity. And again, you at some point here hit the noise signal. It's just that you could have any similarity you find through the alignment could have arisen by chance. Two things that are unrelated look the same in terms of their sequence in the comparison as two things that are not. And that is a big challenge for the alignment field. To intrude further, I told you that up here you do pairwise alignments. You just take two sequences and compare them. Here, somewhere you use sequence against profiles. So you build up a family of related proteins and you know exactly what is important in that family and you align against that profile. And down here, what you really need is profile. Same idea, except for now you compare two families to each other. Comparative modeling works, but because in this particular case I show you 4 different structures, and 3 of those, in fact, are very different from the lead structure. Nevertheless, they look relatively similar. That system, that idea you can use, or that fact you can use to, once you have, for instance, observed the purple and you want to predict for the green, you just look up for the purple. Once you find that target in the database, you look up the purple structure and use that for your plotting. As you see in this particular example here, purple is not exactly green, so you make a mistake. But overall, you're doing fairly well, in particular behind the background that without this comparative modeling, there's nothing else you can do. The most used tools out there are Modeler and Swiss Model. Both that is available for more expert users, download. That is really for anybody in internet web server. And comparative modeling is currently applicable for about 1/3 to 1/2 of all the proteins. As I said, we know 55 million sequences. For about half of them, or 1/3 of them, we can really use comparative modeling to find some region of a known structure that is similar. Okay? Okay. Now, once again, comparative modeling is important because we cannot easily predict structure. Why can we not? In principle, again, the sequence determines structure, determines function. Meaning, you take a stretched out amino acid chain, you throw it into a solvent, water, and you get a unique 3-dimensional structure. So in principle, the folding, the event of taking this stretched out chain, throwing it into a solvent, and adopting a unique 3-dimensional structure, that event is called folding. The folding of that chain. that in principle is well determined, and you should be able to compute it. Before I go on, does anybody in the room know a famous unfolding event that happens to some of you at least once a week, I would guess? Protein unfolding. Have you ever observed protein unfolding?
Well, not for myself, but I've read somewhere that the scientists were able to unfold the proteins of eggs by some experiment and make them unboard. So
yes. So scientists are able to unfold eggs. Thank you for your comment. Sorry that I turned this around. In fact, not only scientists are able. Every single time you boil an egg, then you unfold the proteins. The reason why, when you put the egg into the pan, what happens, in fact, by heating, you unfold the protein. By unfolding the protein, this stuff gets bigger. It unfolds, it adopts. Folding means adopt the minimal volume that you can in water. Unfolding, therefore, means the volume increases. And that's exactly what you see in the egg. When the egg white, for instance, becomes white. This is because the density increases, because the whole thing gets hard, and that, in fact, is because you unfold the protein. Okay? That's a typical unfolding event that many of you have experienced in your daily events. Again, doing it backwards is a little bit more the complication here. In principle, you should be able to do it from first principles, and indeed, there was a publication in the Washington Post in the 60s that claimed the folding problem has been solved. Meaning, we can do it, in fact, in a formula or in a computer. There was another article about 10 years later in the New York Times this time, and that stated the same thing. The folding problem has been solved. There was a third in the 90s in the Washington Post, once again. Again, the folding problem had been solved. And you immediately see that if the problem gets solved three times in a row, at least there was something wrong about the first article, right? Now, arguably, they are all wrong. We have absolutely not solved the problem. The reason why this went wrong, why these journal papers were published, was not because somebody tried to cheat. This was because somebody had convinced themselves that they really had solved the problem. How can that happen? Well, essentially, imagine you write a computer algorithm, some machine learning tool that fits the known structures. And with this machine learning tool, you can in fact predict a new structure by simply doing comparative modeling in a machine learning device. And then you may believe that in fact what you did is you predicted a structure because you can in fact predict it. In reality, what you did is you learned it by heart. In reality, what you did is nothing learning the principle, learning to understand how the folding works. What you learned is how this particular thing adopts a three-dimensional structure with enough free parameters. And now the question, of course, is 3 times over the last 40 or 50 years, in fact, it went so wrong that it went into the big literature, into the big journals. Washington Post and New York Times are the journals in the US. So if you make the headline in these journals, then you really did something that is remarkable. And that is what they did. And that tells you how difficult it was for those groups to avoid that problem. How could we avoid it? What could we do? So what could you do for your work such that a similar problem will not happen to your supervisor? In at least 2 of these cases, in fact, in most of the cases, the work is done by you guys, right, by the young ones. I'm just telling you what to do as a supervisor, and you do the work, so you come with the results. Sometimes it's up to me, and I sometimes can, in fact, tell you you're wrong. But in some cases, we both may fall for the same problem, right? How can we avoid this? What could we do? So given the structure of the room, we cannot sit down in groups, and discuss this. Any ideas? What could we do to, yes?
Applying to already known structures.
Yes, so you said apply, and I'm just taking your ball, because I'd like to somehow create a communication. So applying it to known structures, that is in fact exactly what those guys did. They found a structure, they learned from that structure, and they applied it to a known structure. In fact, in the 60s, there were not many structures around. So the set of those for which they could do it was very, very small. In In the 70s, the second one in the New York Times, in the 90s, the third one, there were 100, at least in the 90s already. So they did it for other structures. That is true. And we need an additional step here. The problem is, yeah?
I think I remember this from the previous lecture.
Then you're not eligible. The idea is that you think, I mean, at least that's what I try to induce in you, is that you use whatever you learned and invent the problem. So if you remember the problem, it's not really inventing it. It's true. You're right, and you're right. You have I suspect that you have the solution right here. In principle, it was a different story. It was a different context, but the story is the same. So again, the question is, how can we? We need to use known structures. You're totally right about that. The problem that happens is that someone develops a method, and that method works only for the proteins that exist today. Checking it on the proteins that exist today, everything looks fine. And it appears as if we understood. How can we avoid that? And in fact, there's a very simple word or terminology in the field of machine learning that addresses exactly that problem. The point, all the data, or these 3 newspaper articles that I talked about here, Washington Post, New York Times, and Washington Post in the 90s, predated machine learning. So part of the answer is really something that machine learning does every day. It's the bread and butter of machine learning in some sense. Does it give you any idea? So again, the question is, how can we avoid to fall into the trap of overestimating the performance of our method? Yeah?
We don't know the structure, so we only use our method, our machine learning method on a small part of the things that we know, and we pretend we don't know the rest solution.
So he says we will present our solution only on a part of of the set that we know, and the rest we pretend not to know and hide, which typically is referred to as cross-validation. The bread and butter of machine learning. Yes. That is absolutely what we have to do. Now, here is another problem. Assume we had 100 proteins. Assume we would just take 50 under the table and we pretend that we don't know them. Okay? So I develop my method for the 50 I have in my, the 5 okay, let's play it with 5 and 5. The 5 I have here, I develop my method on, and the 5 I have here, I hide. Once I have my method, I take these 5 and ask how well they do. Okay? That's the idea of cross-validation. Now here's the additional problem. What if these would be very similar? What if for every one of these 5 here, I also have a thumb, I have a thumb here. So on the thumbs I can do homology modeling. They look almost identical.
You do multiple runs with randomized sets.
Yes. So with randomized sets, that what would happen is that sometimes, so I have my 10 proteins, and sometimes in my randomized sets, then I would have 2 thumbs. And sometimes I would find the thumb. So on average, you say, well, we would still have a large overlap, right? Since by my definition, there's 50-50 overlap. So even a randomized set would still see a relatively good performance, which in fact is comparative modeling. And we want to, so the idea here is, can I understand the way folding works from first principles? Can I understand that by modeling the energy, not by doing comparative modeling? And so what you say would already help me. It would clearly direct or turn my estimate to be a little bit more conservative. But in this example that I give you, 50-50, the problem would not completely go away. How could we make it almost completely go away? Again, the problem now is that in the 2 sets that I have, the one cross-validated that I'm hiding and the one that I'm developing my method on, they are too similar. And you're right, so randomizing would help. But in biology, there's something that is particularly important in biology, is always true in machine learning, and this, whenever you apply machine learning, that's a technology, and that in fact is what he wanted to say, that's where the two things relate. Wanted to say that you have to always use this additional way of caution before you randomize. And that means essentially learn something about the data. What could you do? Mind you. So we're trying to do a method that does something that comparative modeling cannot do. And here we are, that gives the solution away. If in these two sets, random separation or 50-50, no matter how I separate them, if any in the other set can be reached by comparative modeling, then that's the wrong leave-out set, the wrong hiding set. Because for that I can use comparative modeling. And what I want to prove here is that I can do something more than comparative modeling, all right? So anything that can be reached will be thrown out. And that in some sense is clustering, if you want. Some level of clustering. So I have a condition that these two sets have to meet. They have to be, by one particular criterion, more different. Every pair in there has to be more different than, or it cannot be as similar, so similar that comparative modeling can be used. It's a clear definition. It's a clear way to massage or treat your data sets. And then that problem is in fact completely addressed. Now here's another way in which we can, let's call it, even be more sure that we don't cheat ourselves. And that is use structures we haven't seen. How would that work? Well, when you develop a method, you use data. You begin today. You extract your data from the database. That takes you some time. Then you work on your method. That takes you another time. Say the whole process takes 6 months or 4 months or 3 months or something like that. And now you have the method that you really believe in that cross-validation test works well. Over those 3 months, new structures have been added. All you need to do now is find in these new structures one or a few or some reasonable sized set that you could not treat by comparative modeling from whatever you had as a knowledge when you started your project. And these are the new ones, and they in fact simulate what will happen in the next year. That's an additional way of doing it. And that is in fact what the critical assessment of structure prediction, the CASP meetings do. April to May, the organizers collect experimental structures that in fact, essentially they ask experimentalists, are you going to solve a structure over the next months? If so, give me the sequence. Before you know the structure, we're going to use these sequences and send it out to people who predict structures. It's the prediction season is then June, August. The deadline of the prediction is before the structure is published or sent out for publication. So as long as nobody in the world knows the structure, people can predict. In September, November, assessors look at these structures that then came in, the predictions, and then there is a meeting in December. And that has been going on for many years. It started in 1994. In fact, it started just after this Washington Post article that I talked about, and it was in fact spawned by that Washington Post article, by the shock that somebody can be fooled into believing that they solved it again. That's the number of groups that participated here only in the first 10 years, is still going on. It is a biannual meeting, and the number of groups is increasing. The number of submissions is increasing. One major outcome is that in fact only homology modeling is reliably predicting structure. The other one is there is just no general prediction from 3D structure from sequence alone. We cannot do the loop modeling that I talked about previously for long loops. We cannot model new structures. All we can do is find better ways to map things for which we don't know the structure and have sequences into structures. And we're going, over the years what happened is that this became better and better and better. It becomes better because we're getting better at recognizing the similarities. We're getting better at, in fact, then modeling it in and wobbling around. But essentially, this is still all in principle the idea of comparative modeling. We cannot model from first principles, except for very short fragments, and then in particular contexts. What happened over the years is that more and more meta servers were created. This started simply in 1994, when the first CASP meeting happened. Most scientists came and had done their own software, and they had never used each other's stuff. Some came away from the first meeting as pseudo-winners, or did better in some things than others. And whoever saw that somebody else has something better and that it will allow me to win took that bit for the next CASP meeting. And that got into the evolution of meta servers. So servers that simply integrate as many tools as possible and try to find the best solution out of it. What this one shows here, the color scheme is over the years. This is target difficulty, this is the degree of accuracy, where right is up here. And the different colored lines show you that over the years there is a slight improvement. Some targets are very difficult, some targets are very easy. And essentially we do better over the time, over the easy ones up here, over all the time. So all of these 10 years of CASP are essentially at the same points here roughly. And here in the hard ones we do a little bit better later. So we move from here to there. But essentially we still are way below what we can do for the easy targets. And the easy targets is simply defined as comparative modeling. Problems with CASP are, first of all, that we have a very limited number of proteins. And now here comes another one. That's the first problem. So I said, in order to do this trick that you look up a new protein, you have to have numbers that are meaningful. And 100 proteins is a number that is rather small. Because many of these 100 proteins, as I showed you, are easy targets. So this is comparative modeling, we already know it works. What we care for more are the complicated ones that really look different from anything we have seen. And every CASP meeting we have just a bunch of them, a handful. And that is not enough to make clear statements. Here's another problem. Say we have the situation in which method uses 53 of these hundreds and gets a performance of X. And method B uses 35 and gets X plus Y. The question is B better? So Y is a positive number in this particular case. So we don't know. Indeed, X plus Y by my definition is larger than X. So in that sense, numerically the number is higher, but it doesn't use the same data set. So the most extreme one, the most extreme solution of this, I'm going to present something to you that a very intelligent person presented to me. So here is the performance of the method. Higher means better. Here I show just a distribution. Asked how does his method perform for a bunch of proteins, he said like this. If I presented that to you, react to that.
Y-axis is the number of proteins?
So what I show here, yes, the y-axis is the number of proteins, so that's the distribution, how often am I getting a certain level, and that here is the performance, the accuracy, where this is higher. Say this is higher, 100% for whatever that means, 100% right. Okay? And say in my graph this is 50%, and this is the curve. So I'm just doing the same curve 3 or 4 times. That's the curve he gives. Anybody jumping at me? And mind you, this was a very clever guy who had worked as a scientist for many years. So it's not that obvious, obviously. Yeah?
I think it's not very good because it reaches the end, the 100% accuracy, when the normal distribution is very low.
Okay, so let me repeat for the people in the back of the room. The statement is, this method is not very good because for a lot of the data here, 100% is not reached. And this has a lot to do with talking about how well computational biology does. And I'm going to pull this tooth. Computational biology is not like engineering. We don't get the level of accuracy of the speed of light. So I will show you many, many, many graphs in which this is far from reality because the performance of methods is way worse. You're right in your observation, but this field typically is not that low. Yeah?
Maybe I should repeat my sentence again. So my idea was it only reaches better performance when the distribution is lower. So
yeah. So you say this is relatively few that have a very high performance. But again, so what I'm trying to say, for many methods that I will present to you, 50% is an amazingly good number. That's what I'm trying to say. I'm sorry. I didn't understand what you meant. So it's a good point, and this is a point, it's a totally right reaction. In fact, I should have made that before. So welcome to the field. Most of the time we don't get these curves. So this is a totally invented curve, and whatever, I'm going to show you curves in 2 weeks from, or in a week from now, like that, and you will see that this is not that bad. Well, okay, the ones that I begin with look a little bit better, but that's one comment. Okay, so it could be better, sure. Any other comment? There's something wrong here. Is anybody stumbling over it? Something wrong, not on the level of where these numbers actually are. Is there anything that clashes with the shape of the curve? What does the curve remind you of? Can anybody throw a normal distribution? Does it look like a normal distribution? Ah, exactly. So there was a student in the room. I just stood there and I didn't even know what to say when I saw that. And the student simply asked, okay, it looks like a Gaussian to me, like a normal distribution. Where's the other half? You know what he said? Oh, he said, those proteins are performing very poorly. I threw them away because my method doesn't work for those. I only give you the results for the ones where the method works. Is that right? Why not?
Because it's the same thing when you say, I will show you this, but I won't show you that just because I don't want to, because otherwise you will say that nothing works and then I won't be able to...
You're hiding data from people, and it is like just showing the good part of it. It's true. That's one way of saying it, and then if you interpret this statement like this, then you would say this is cheating. Let's call it by its real name. However, he could have argued, wait a minute, this is not cheating. My method, for some proteins it works, and for some proteins it doesn't. I can tell you which ones those are. I can tell you, okay, guys, for this half here, for these 50 proteins, I'm not going to do a prediction because I can't do them. And for the ones that I give you, this is the distribution. You're still not happy. Why?
Make no sense. Well, yes, of course, you can predict for one part, but you cannot predict for the other. But then that should have been stated in the paper.
That's a very good point. So the crucial point here is you have to state that. You have to show the entire distribution in your paper, or whoever you talk to, and then you have to prove that before you observe the distribution, you can actually predict which one is which for the proteins that come in. If you then can also prove to me that in fact you can, before you see the result, when you only look at the sequence, you can know whether your protein will be there or there. If you can prove that too, then you can say, okay, from now on I'm going to compute my values on that set. That would be okay procedure. You're totally right. That brings to the next issue. So the first one is the question, is 100 proteins enough? The second problem, the second question is why enough? Remember, we had this thing that method B had for 35 proteins, X plus Y. And that is the story. Method A for 53 cases and method B 35 cases. In this particular example here, we have an example where the method in fact decides only to use 35. Before the effect, they don't know the answer. And they say we use only those 35. So they know something about their method in that particular case, okay? And that is another problem. We have to rank correctly. Inappropriate ranking is very complicated, brings wrong conclusions. Now, there are too few targets at CASP. And here is a question that I pose. I'm going to answer that question, but with a slide that you will not understand. So you will have to trust me that the next slide says what I say it says. The question is, say this really happened in one particular year of CASP. We had 29 proteins that were new, that we could use. But some methods only predicted some of them. Only 11 of these 29 were predicted by all methods. I want to compare a bunch of methods. In fact, can't remember, the number of methods are 8. So we have 8 different methods. And should I now use for every method whatever they use for the prediction, which is a subset of the 29, or should I compare them on the identical set of 11, which is a tiny number? What is better? (11) identical, 29 any. In this particular case, it's really 29, so the total set was larger than 29.
What did we take away the 11?
Say it again?
Why did we take away the 11 from the 29?
I do not know the answer to that question, but we will get back to that later. So that's, we keep it. So 18. But the question that I pose to the room, what is better for estimating the true performance of a method? Taking 11 identical, small number. I already know that 100 is not enough, 11 is smaller. Or taking 29 any. Okay, let's just have a raise of hands. Who believes this is the best? And so you have to say one of the two. No opinion doesn't count. 29. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13. Identical. 11 identical. 1. Oh, that one is easy. But the sum is not, in the room here, the sum is not 14. Okay, fair enough. That's as good as we get. And this, in fact, I can tell you, crowdsourcing really is an amazing way of doing things. This is the way the whole field saw it. And this is for many, many years why the field did exactly the 29 any. And it is wrong. The one in the room is right. I told you already, this is a complicated experiment. This is results for 29 identical, 29 different, and 11 identical. So what I do now is I have a large data set and I simulate CASP. I simulate CASP means every year I know I have 29 and I just pull 29 out of my data set. And then I rank my 8 methods based on that, and I used 11 identical and ranked my methods on that. In the background I actually have 1,000 proteins. So I know the true rank of these 8 methods. I have 1,000 proteins for which they all work the same. Okay? And then I can simply look at the performance of the 8 different methods that I have here. And those different symbols are the different 8 methods. And this is essentially number of years of random draws. So number of years of running CASP in some sense. And then I ask, is the best method best? If the best method is best, then that's an okay ranking. And what we see here is in the 29 different proteins, every method in some year gets best. Even the clearly worst method. This is a little bit not as clear because 11 is just a small number, but the worst method becomes the best in 1 year never happened in the 11 identical case. So the ranking of this one here is much more stable than the ranking of that one, although it very often does not produce the right results because 11 is just too small a number. But given that 11 is too small a number that is almost triple that size, 29, with different proteins is worse. So never do apples and oranges, even if you have very, very, very few fruits. When you compare things, apples with apples and oranges with oranges. Another way of doing that is by having pairwise comparisons. In this particular case here, what I do is I have 4 different methods, and I simply ask their diagonal is grayed out because method A is method A. So now I ask, is method A significantly better than B, C, D? Significantly better means dark blue. This translates to significantly worse. And the coloring is, as you see, already indicating in this chosen example, it's not completely stable. I believe I made a mistake in the coloring. So this should be light red typically. So now, this way you can build up a table in which for every pair comparison, you can use an identical set that is as large as possible. So what's the problem here? The problem is if you have 8 different methods, there are different reasons why the methods don't predict. Sometimes the reasons could be like here. But the author of the method knows that my method will not work for that protein. But more often than that, something goes wrong. The server goes down, the experiment is running for 3 months. So for 3 months your computers have to be up completely. So at some point the query comes, and if your server happens to be down because there's an electricity shutdown or something like that, then your protein, this protein will not participate in the competition. And since we have 8 different methods at 8 different servers at 8 different places in the world, the reason why they fall out may be very different. So finding a common subset for 8 methods or even more methods is increasingly difficult. And in fact, I showed you that 300 groups participate. You can immediately see it will get more difficult the more methods you have. Doing this pairwise comparison, for every pair of methods, the probability that you get a high number is higher. Okay, now once you have this table built up, we get into the next issue. How do we translate that into a ranking? Any idea? So essentially, what we see in this matrix is that A performs significantly better than B, and so forth and so on. How do you now rank? What's the best method? In this table, is it clear what is the best method? Ready? Yeah, A or D, right? A sounds like a good idea, but D could be the runner-up to it. There is some indication that A may be better than D, but this may also be not significant, according to your criterion of significance. Say it would remain as wide as that. Then we could not distinguish between A and D, could we? But the next question really is, how do I formalize that? How do I get the matrix into something that is a rank? Can you think about an algorithm to do that?
It looks to me like a schedule of football championship. You just give 3 points to one winning in each of the comparison, and then you have a result.
That's a great idea. So the idea is you give out points, whether or not you're a winner in a game, in this particular case, in a head-to-head comparison, and then you simply add up these points. Is that going to give you a unique answer? My question to you. So you understand the algorithm, right? Essentially, you count the dark blues or the dark reds, or so points where you, in football we would have in fact 3 different classes. You lose, you win, you draw. Draw would be you cannot distinguish. A and D would be a draw in this. So a draw gives 1 point, a win gives 3 points, and a lose gives either 0 or minus points. Is that a clear solution? The problem is in this, for the 4, that pretty much actually models what you see. But once you have a very large matrix, which particular numbers you put down, whether the win is 4 and the draw is 1, or the win is 4 and the draw is 2 or something, may influence the outcome. And that again is something that you have to sort of find a way to simulate. How could you simulate that? How could you simulate, yeah?
I think we need to assign points based on significance. So is it a significant
Yes.
Improvement
Yes.
A to D? If yes, we assign more points.
Yes, but oh, you mean a point that is proportional to the number, to the degree of significance? That would be, yes, that would clearly be an improvement of of the simple idea of draw, win, lose. But it still would not get rid strictly of the main problem, which is that the relative value of the rankings, or the points you give, may determine the outcome. How could you, so it may determine, it may not. How can you find out whether it determines the outcome or not? You're computer guys, what can you do? You just simulate. You just give a bunch of scoring schemes, bunch of different numbers to use, and then you run the same matrix again and again with different numbers. And you see how stable the ranking is. If the ranking depends on your choice of the numbers, you have a problem, and we need a new method. If it doesn't depend, you declare win. Then you will say, okay, I used the football ranking because it doesn't matter. I tried different rankings and it was all the same. If the ranking does matter, then you have to look in more detail, how much does it matter? Are there statements you can make? Can you make the statements that, for instance, in this particular example here, A and D are best? Would that be beyond that? Would that be true for all the numbers you put, for all the scores you can put? If so, then that is a statement. And that's another way of, in fact, deriving a ranking scheme that is stable from the raw data. The solution you find is the only solution that is compatible with the idea that you want a solution that is not depending on artificial things such as your free parameters, right? And that's easy to test in a case like that. What I want to induce here in you is, once you have, and it's not easy to get there. And in fact, in this particular problem here for the CASP field, it took over 10 years to turn the field around and in fact implement methods like that. And to then, from that implementation, to get to the next step and in fact come up with an automated score, it still has not quite happened. There are also always social components about things like that. But as long as it's in a computer, it's easy. But what I want to induce here in you is, once you come to this point of this matrix, try to play around with it. You see, in this particular example, you see A and D stand out. Don't stop there. Do the next step and put in your algorithm, in a new algorithm, into your computer, what you believe you see. Test your data, okay? Okay, the solution of comparative modeling. Comparative modeling is the most accurate way to predict structures. Essentially, it cannot get better than the template, so the protein that you find is similar. If that structure really is different, then you cannot solve the problem. So you can get as good as the similarity between the template and the target. So the one for which you look up the structure experimentally and the one for which you want to predict the structure. Mostly it's driven by the accuracy of the alignment. So when you make any mistake in what you believe is the corresponding point in 3D, it's difficult to retrieve from that. And loop modeling is still a complicated story. So loop modeling means that I have 2 points that are constrained from the structure that I observed. In the structure that I want to predict, or the protein for which I want to predict the structure, there is no connection observed. So I have to make it up. And that is loop modeling. So finding a new loop, a new region between 2 constraints, and that is still unsolved. Side chains, essentially so far we talked about main chain only. We talked about one amino acid, one residue in a protein has one essential central point. And that's the point we have looked at. Side chains are what makes the colors different between the different amino acids. How are they placed? That's yet another problem. Okay, let's talk about how we get to 3D structures from experiments. And again, so that is the level of detail of one representation, 1 point per amino acid, per residue. Again, amino acids when put into a protein are called residues. And that is a cartoon. How do we do that? Currently, or this number is slightly outdated, but roughly, the protein data bank collects 3D coordinates of proteins. The date is missing here. This must be 2013. So today is over 100,000, the total number here. Then it was 84,000, and that was about 1% of all the proteins. So for 1% of all the proteins, we know the three-dimensional structure, and today the number is smaller because the number of sequences grows much faster than the number of structures. X-ray is 75,000, so 89% of all the known structures are from X-ray. So percent here means percentage of all the proteins, percent from below this line, and I should visually separate that, means the percentage of this number. X-ray, NMR, EM. Let's begin with X-ray. You take the protein out of its natural environment, or you express it in a way that you can grow a crystal. What does it mean to grow a crystal? You have to have a lot of this material. You have to have it at a high density. And in this high density, you have to, in fact, grow a crystal. Have the conditions such that you will find a repeated unit of the same thing again and again and again. That's what a crystal is. This is not easy. It is not easy to get that amount of protein purified. And it's not easy to, in fact, find the right conditions under which they crystallize. Once you have, however, the crystal, and protein crystals, this is a protein crystal that I showed there. It's from Wikipedia. When you see talks from crystallographers, they always show you their crystal because they are beautiful and they're proud of them. And in this crystal, when they look at this, then they will already see how good it is. I don't. For me, it's just a nice crystal. What you then do is you put X-ray beams into that crystal. And that again goes back to what you see is the same experiment that is shown in the subway down there, here in Garching when you arrive with the U6. You do the simple scattering of X-ray beams on the atoms, and that allows you, the refraction allows you to find out where the atoms sit, where the electron density map sits, and that allows you to build an atomic model. Any idea how long that takes, what that costs, how easy that is? Does it strike you as something that is simple? So everything that biology does is in fact highly non-trivial. They can do a lot with gene manipulation, but it always started with something that is not easy. Typically, machines are easier to handle than life. So just getting one particular protein into your test tube is in fact a problem. You can do this now by robots for some proteins, but not for all. We do not have all the human proteins in a tube, for instance. But again, is this something that is easy? Now, I'm going to throw a few numbers. The whole process to get an image of a protein, I showed you several images. Get an image of a protein takes, is the question, 1 month, 1 week, 1 week, month, 1 year, more. What do you believe? Who believes it takes a week? Who believes it takes a month? So there's some count here. Who believes it's a year? That count is higher, but not much higher. Who believes it takes more? That count is existing. Most people didn't count. Most people didn't give an opinion. So when I started, it was in fact a PhD project to do one structure. And some PhDs were given without actually completing the structure, meaning that it took a PhD project in the US is 5 years. So that used to be that ballpark of time. 1 week is unrealistic. Today there are examples where we can do it in a month and in a year, and some still take more. A year, somewhere between a month and a year is a good time. But this only works for some proteins. Most human proteins, in fact, have not been put through this procedure successfully. Just a tiny fraction of the human proteins. And we will get back to that. It doesn't work for everything, and that is in fact the reason why for most proteins we don't know the structure. So that's one thing, and it takes a long time, and it's not possible for everything. Yes?
Why do we need to put them in a crystal to do the X-ray?
Because so if you had this protein in a solid, essentially assume you had a cell, and you shoot X-rays onto the cell. The problem there is that the protein moves. The phasing here works only because the crystal, in fact what happens is you get many copies of the same protein, and you get the same atom in all versions of these copies in the crystal sends the same signal because they are frozen in a crystal. If you put them in solvent, that no longer is the case. And you cannot, essentially you will instead of getting scattering data that allows you to retrace from the scattering the origins of the atoms, you will just get a mesh of data that will not allow you to do anything. That's the problem. In fact, proteins in the cell move so much that we absolutely could not do that. Even if we had, essentially imagine we could pin them. We cannot, but imagine we could pin them. Even then, they would still wobble so much that this would not work. We really need something that is as solid as a crystal in order to do the phasing. But here's an idea for something that you have all encountered that does work for living material, which is NMR, nuclear magnetic resonance. So in an NMR, you simply take it in a solvent. You go into these tubes. Some of you have at least seen them, where you lie down in these nice tubes and the world is shut off you. And that does living material, because essentially what it does, it induces with a magnetic field rays of the protons. And the response of the protons to the magnetic field is what you measure, and is what you then use to deduce the structure. Now for proteins, these magnetic fields are not as small as the ones that you see in hospitals. So this is not even the biggest machine there is, a 900 megahertz machine. The person is up here. So this is 3 stories high. This is in City College, the New York Structure Center. There are several centers like this in the world, where you just have a bunch of big NMR machines, essentially big magnets. And building a room like that is highly non-trivial, because you need a lot about static. This room has to be shielded. And you don't want to walk into this room with your credit card or anything like that. Anything that you carry that has a magnetic stripe will completely be wiped in this room. This is just high density of magnets. And then what you get essentially is something like this. Now in this particular case here, what you see is that there's no one solution. You see that there are in fact several solutions here. And that is because you create a model that is compatible with the data that you observe. And in some places, the different models that you do, you simply do a simulation. You have a 2D map. The 2D map defines the 3D structure. 2D map because you look at similarities between proton responding to the magnet. So you get 2D constraints. And from that 2D map, you compute the 3D map. Unfortunately, it's not, you don't get the real 2D map. You get local 2D maps. And that means that you can actually compute different models that are compatible with the data you have, with the experimental data. In some places here, all the models essentially come to the same point. In some places, however, different models give very different solutions. And this could be, and that's in fact the NMR interpretation, this could be because we have motion. It could be that the protein actually moves here. What we have here is something where the protein doesn't move. And here we have functionally important movement of the protein. In this particular case, it's at the ends of the proteins, and there we observe that very often. Overall numbers, NMR is available for about 10% of the proteins for which we know 3D structures. NMR is easier because we don't need to grow a crystal. It's more complicated because, in fact, just a sec, getting from the protein to the final solution takes experts and is very difficult to get from the experimental data to the final solution, and it cannot be done on very long proteins. So NMR can be done on proteins that are about 200 residues long. Most proteins are much longer than that. That's the second problem. Yeah?
Could you repeat please how it works exactly, a little bit what the magnetic field does to the protein and how it's useful?
What I understood is, could you please repeat?
Yeah,
But then I never understood the rest.
what does the magnetic field do?
Oh, the magnetic field essentially excites the protons. Because the protons will align in the magnetic field. Through the alignment in the magnetic field, in fact, they will align differently when they are near another proton. And from that change in the local magnetic field, you essentially get proximities. C alpha, the proximities. And from those proximities, you build these models. In fact, phasing for X-ray crystallography or getting from the raw magnetic data to the structure is a long way. Both received many Nobel Prizes. And the fact that this is a few years old does not mean it's simple. So what I'm telling you is an idea. I'm not actually explaining how it works. Let me make that clear to you. Okay, here's another way in which we could do it. So we have a crystal, we have something in the solution. Why not look at it with a microscope? It's the next thing you could ask. And the answer to that, again, is twofold. First, the size of proteins, they're a little bit too small for microscopes. Even electron microscopes, cryo-EM, so you freeze the material, cool down the material, and then you do electron microscopy on that. Even for that, they are still a little bit too small. What you get, what this one here tells you is the angstrom resolution. For 32 angstrom resolution of that protein, in 4 angstrom it looks like this, in 32 it would look like this. In 16 like this, and these in fact these 4 are typical images that you get from EM. So this is already better. I believe at some point a few years ago the average accuracy of EM was more in this ballpark. So this is sort of the newer ways of getting at it. And the 4 angstrom model, what you see is you don't see the atom. You don't see the protein. What you see is a surface. And in that surface, well, sometimes you see holes. Actually, here you don't. And many of the EM solutions were like that. Here you begin to see holes. Here you do see more. But you still don't quite see the atom resolution of a structure. You're not getting the same information. But you could possibly imagine that you now can combine that, and that's a paper from David Baker, not David Baker, from Ju Chu, using cryo-EM to combining cryo-EM and comparative modeling and modeling processes to combine both. Now, if that is the image you have, and maybe some parts of the proteins you can model, then your task will be put these local predictions into that EM and see whether if you predict this model, would that fit to the overall surface shape that you see. So in this particular case, you combine EM to give you a surface constraint with local modeling, comparative modeling, and putting things together in a comparative modeling. It's a great way. Unfortunately, still the problem is that if EM data is too small, you have too many ways of putting it in. But this is one of the major lines of research these days, that you try to, in fact, combine modeling with experimental technology, because EM is just so much simpler. You don't need a big magnet, you don't need a crystal, you just freeze it a little bit and you look at it with a microscope. That may be the solution, certainly for larger aggregates of proteins. On the level of a structure, and that's from X-ray crystallography here, at a resolution, so at an accuracy, at a resolution of 1 angstrom, that's a high resolution, 3 angstrom is a low resolution. Well, there's still, remember I told you EM gets best to 4, so this is still way better than that. So for 1, this is an aromatic ring. It's a side chain with an aromatic ring. At 1 angstrom resolution, you absolutely see the ring. For the 3, you may see the light, the blue mesh here. The blue mesh essentially describes the experimental data. At 3 angstrom, the ring can be put into different places. And there's some optimization in which you, in fact, can fit the green to the background blue mesh here. Well, here is absolutely clear. The blue mesh now is no longer a mesh. The blue mesh is something that is around atoms. Okay? And that's the difference in resolution. This resolution is great for certain biochemical studies. Drug design clearly wants to be on the side of this or smaller. Whenever you develop a drug, you try to get crystals that are this and better. There is no drug has been developed without having a crystal. So drug design, making new drugs, always begins with knowing the 3-dimensional structure of the target. Okay, again, 1D, 2D, 3D. 3D, I'll get back to it at the later part of the lecture, but now I'm going to say, assume we cannot do 3D. What simpler descriptors of proteins do we have? And we already know secondary structure, which in fact is stabilized by hydrogen bonds. That concept was introduced by Linus Pauling, who got a Nobel Prize for it. And the hydrogen bond formation and the helix here is between I and I plus 3, I and I plus 3, and so forth and so on. For the beta strand, is between, every 2 goes to the next strand, neighboring strand. And that is in fact in cartoons, you can see these helices and strands. That brings us to the next question. When was the first, so I'm showing you these images. Do you have any idea when the first structure was done? Anybody believes it was 1800? Okay, no, there was no X-ray crystallography. Yeah?
Was that the one made like
The one that is made?
I've seen some printing structures that were made in
Hair.
just made by hand.
Oh, built by hand. Actually, so we get back to that point later. No. Yes, so there were many attempts to predict how structures or proteins would look. And indeed, it is also true, somehow your hands induced in me this idea, so EM pictures of hair or of, in fact, silk, those are examples where you essentially see the same protein again and again and again, and in some sense you could call that a protein structure. But the first atomic resolution really was at the end of the 50s, Kendrew and Perutz, 2 Nobel Prizes, and those, in fact, both in Cambridge, 2 competing labs. This one was a little bit earlier than that one, and those are the 2 structures they got. Myoglobin and hemoglobin, both are important for the blood, for oxygen transport. Anybody notices anything? By now you have seen maybe enough structures.
There are no beta strands.
Exactly. So in fact it took another 10 years before the world had 4 proteins. So in 1917 there were 4 proteins known, and they all were all alpha. So the conclusion was there is only alpha. Yeah, it's easy to smile about that today. And that gets us back to Pauling. Pauling is here. Pauling really had in fact published a bunch of papers. And in this bunch of papers, he had by pure ideas about how an atom bond is done and how that could lead to aggregation of larger groups. 51, 53, he had predicted alpha helices and beta strands in a set of PNAS papers that got him the Nobel Prize. He had predicted all kinds of things in these papers that never ever were observed. So in the 70s, the conclusion of saying, okay, he published so many things that were wrong. One was right and that got him the Nobel Prize, so that's fair. But this is also wrong, they then said. 8 are wrong, or 9 could be wrong. It's not a big step, right? From what I showed you immediately, this is clear, is not the case. There are different methods to predict, to get from secondary structure from 3D to secondary structure. So essentially, the question is, how do I get from that to that? And because this essentially is only an annotation, it's a cartoon of that, meaning, how do I really get to the annotation? How do I get to the annotation? What is a helix? How to annotate 1D secondary structure from 3D coordinates. And again, the idea was to have a discussion about that, but the way you sit, this is complicated. How could you do it? So I give you 3D coordinates. And what I want is something that looks, like that. An assignment of where is the helix, where is the strand. How could you do that? Any idea? And doesn't it jump at you once you have assigned it? Come on, nobody will tell me that this thing here looks the same as, the red looks the same as the yellow, right? It looks different. Look different, you can translate, you're a computer scientist. Anything that looks different you can translate into an algorithm.
Can I go through the sequence of atoms and check if the coordinates for one atom is, for example, after the, 100 certain amount of atoms, it starts to repeat itself as a helix?
Yes. So, repeat itself as a helix. So essentially, the way you could define that, I assume what you mean is, you look at a certain geometric shape, and you see whether that certain geometric shape is again and again and again. Yes, it is even simpler. Here I have a helix. A helix, I said, is stabilized by a hydrogen bond. There's I and I plus 4. So you could even in a computer or on a drawing board draw a helix, and then simply ask, where do I find that in my structure? Have a match or an ideal idealized helix as a mask. Run the mask over your 3D coordinates and say bingo whenever you find something that looks exactly like the mask, right? Now, once you find the mask, what you still have to do is to extend it. Because some of these helices here are not as ideal. They're short, they're a little bit curved, they're a little bit pulled. So there are deviations from the ideal mask, and that is something that you have to handle. But in principle, you could do it geometrically. And in fact, that is what Define does. One of the first methods out there is described exactly the way in principle we just said. You find the mask, you do it from beta strands and alpha helices, and at some point you cannot do it. So some of them are curved, twisted too much, and you cannot recognize the ideal shape, and you say you can't. And then you look at it as an expert and maybe assign the others. From what I showed you, there's another solution that you could have gone. Any other idea? So that was a simple idea, and the simple idea exactly led to one of the first methods that did it, that solved this problem. Any other idea? What stabilizes secondary structure? Remember what I said? Remember what Pauling's idea was, or the Nobel Prize was for? Yes. Secondary structure is stabilized by hydrogen bond formation. In a helix, it's I and I plus 4. That's the repetition you had in your mind when you came up with your first algorithm. And a beta strand is the hydrogen bond formation between adjacent strands. Why not use that? Why not simply look at a bunch of hydrogen bonds? The way we get them is we have a simple Coulomb energy model. And DSSP, in fact, is the method that does it. The dictionary of secondary structure of proteins. So you begin with a hydrogen model, Coulomb model. In this Coulomb model, you essentially say when the hydrogen bond forms. And then, according to whatever 3D coordinates you look up, you ask whether any pair is within some proximity, because the Coulomb model gives you an estimate in kcal and in energy. So if they are closer than that, we say the energy is right for a hydrogen bond. And then you simply look up at the pattern of hydrogen bonds here and in the beta sheet. That is essentially exactly the idea. And in this idea, DSSP assigns 7 states. This is the detail of the 7 states. So there are different helices. I and I plus 4 is the typical, what is called the alpha helix. This is so-called 310 helix. Whereas I plus 3, then there are slightly different types of helices. Overall, this is the word that I used. There are 7 DSSP states that relate to somehow 3 major things: helix, strand, other. Often called loop. In loop modeling, I call it loop. Sometimes I call it other. Essentially, whatever word you use, the fact is helix is something that is well defined. Strand is something that is well defined. And the other state is not defined. Everything else. There are other ideas. So we could also, here is an idea of stride that uses machine learning, that says, well, let's just look at experts that assign secondary structure and learn it from these 3D coordinates. And a more, stride is not, it comes from Dimitri Frischmann, who's in Weihenstephan, is not a pure machine learning device, but it fits into what experts learned. Predictability, here is the idea that I assign secondary structure according to what I can actually predict. Most secondary structure method assignments mostly agree. That's what this triangle says. So, but so in the extreme examples here, we have 3 different methods. Most of the time they agree. All 3 methods say either it is alpha, 23, beta, 13, 23 plus 13 is 36, plus 27. So there you have the majority of the assignments where 3 methods agree. Today, most people use DSSP, which essentially uses the Pauling idea. And it's one of those methods that is around since '83 and is essentially used every day. With that, I believe I'm ready to close the lecture. Is there any question for today? Let me repeat the statement that I very much suggest you go to the exercises, because I do understand that a lot of the details, the level of detail that will be queried in the exam is not the level of detail that I give in the lecture fully. So part of that is given in the exam. That is the incentive to go, in the exercises, that's the incentive to go there. One other issue, I can say it again, it didn't work in the past. We can switch off. Thank you, Colin.